All right. Good evening, everyone. Thank you for joining us tonight. We are honored to have Mary Sand, who is the director of the Cosumnes River Preserve Native Nursery. And tonight she's going to be talking about conducting restoration and growing plants for that effort. Mary has an active foundation of community building, art, design, and most recently working in habitat restoration as a botanical specialist at the Cosumnes River Preserve. Her first contract began in fall of 2024, and she designed and built a low-cost and adaptable propagation nursery and trained over 30 volunteers on how to collect and propagate cuttings of native plants. Mary's methods are rooted in curiosity and experimentation with larger research questions that challenge current land management practices and herbicide use, while examining potential alternatives and circular strategies for control of invasives and revegetation. So thank you, Mary, for joining us tonight, and excited to hear your presentation.
Thank you so much. I'm going to go ahead and just share my screen, start us off. That's weird. That's very strange. Okay, bit by bit. Wow, this is unexpected. It literally just came up 2 seconds ago.
It worked perfectly when we were in practice session.
Yeah.
This is how things go.
Let me stop sharing real quick and get this sorted out. This is why we're plant people and we don't work in the tech industry, right?
Yes.
Okay, we are up and I will return to share my screen. All right, here we go. So thank you again for having me. It's an honor to be here with CNPS. Today I'm going to talk a bit about our nursery project out at the Cosumnes River Preserve, the community-based restoration that we're doing involving volunteers and multi-agency staff to help restore habitat and collect local genetics in order to do that. So here is an overview of my talk. I'm going to give some intro context, then I'll get into some of the vegetative propagation, the wildland collections, which includes some mapping, tracking, and the genetic diversity, collection methods, prepping and potting, some species-specific findings, a bit of the infrastructure we built out for this, and some future directions. So the Cosumnes River Preserve is known for its stunning sunsets and migratory waterfowl and the beautiful murmurations that those birds create in the skies. And it's unique to this landscape because it has seasonal floodplains. And this also meant that we had to consider this while accessing different land for collections. Also, many of the same plants that we grow here are also growing here in Sacramento along the American River. The Cosumnes River Preserve is over 50,000 acres of wetlands, riparian forests, seasonal floodplains, vernal pools, grasslands, and agriculture land. Um, it's a privilege to get to access this land because most of it's not open to the public. So this project provides an opportunity for volunteers to get to explore some of the wild land that they otherwise would not have access to. This land was the traditional lands of the Plains Miwok, and the Wilton Rancheria Miwok is actually a partner of the preserve. There's also a dozen of other tribes recognized, including the Cosumne and the Mokelumne tribes that these rivers were named by. And there's also a breadth of immigrant histories in the nearby river towns in the Delta. I also have a personal connection to this land. I grew up in the town of Locke. So the little pink triangle on this map is where my mom lives and where I'm from. And the green dot is where our on-site nursery is. The map on the right-hand side is showing the various agencies that own and manage this land currently. So there's a combination of state, federal, nonprofits, and NGOs managing this land collaboratively. And this specific project began development just a year ago. Our mission for this project is to develop methods, infrastructure, and best management practices for an on-site nursery where wildland collected plants are grown to supply restoration efforts at the preserve. Our long-term goals include revegetating native habitat and outcompeting invasive plants through adaptive management strategies, and also widening our network of volunteers and partners. And in large part, a lot of the value of this project is in citizen science, is getting to invite people out and participate in this project. This is a little couple of slides about me. My background is in art and design. So I've been working with plants for a long time in a much different context. I've been working with dried and preserved materials, for probably a decade now. And just in this past year, I've started to grow them as well. So this is just a small sample of some of my artwork. And most recently, on the right-hand side, I make these seed bank bugs, is what I call them. They're little critters made of little collected parts of plants. And on a more philosophical level, a lot of my work explores having a relationship with an altered landscape. And I also do a lot of community work every weekend at the Red Barn Collective, which is located at the Folsom Boulevard Flea Market, and it's open to the public. So you guys are welcome to come visit us. It would be cool. I always love crossing communities. And I also have created these specific logos for environmental organizations. This is just some of the background and how I've gotten kind of a foot in the door in the restoration world. And then here is the more directly relevant experience that I've had working in the field. I worked with Hedgerow Farms and California Native Grasslands Association, and then the BLM Nature Conservancy and American Conservation Corps experience are direct partners in this project. Also, the literature that I sourced to inform this work started with CNPS. I was given access to the propagation protocol spreadsheet by a contributor, and so those are the protocols that we use to inform all of the species specific methods. And then I used 2 nursery manuals that were written by the USDA and BLM. Okay, so getting into vegetative propagation. Vegetative propagation, as I'm sure you all know, is clonal growing. So it might not seem like the first to consider when doing restoration work, because it does preserve clones within the genetic diversity that is grown out at the nursery. But this is just something that is one of the limitations that we've worked with to develop protocols to account for in order to maintain as much genetic diversity as possible. We did not have very much of a budget for this project, which is also part of the reason we did mostly just vegetative propagation. It's a much lower cost, and it doesn't require like a full greenhouse setup. It's a much simpler process. So that's part of the reason we stuck with vegetative propagation. So these photos are our nursery. This is actually two sides of the nursery that are separated by the retaining wall that you can kind of see in these photos. The photo on the left is showing new cuttings that are just in their establishment rooting phase, which is it's a process done in the shade. And on the other side, this is like once those plants have been growing out and they've fully rooted, we moved them to the other side and set up shade structure that allowed more light in so that they could establish more. Also, the bins in this left-hand photo are our version of a greenhouse. So they're like really low budget way of maintaining humidity for certain species that require that for the initial establishment phase. So our nursery, since it's out at the preserve, it ended up being a spot where other critters took refuge. So here's some examples of little animals that we found there taking habitat in the nursery. I apologize if some of these identifications are not correct. Yeah, so if anyone has corrections there for me, feel free to call me out. And then these bird species, I never was quite able to identify them. So yeah, let me know if you have some insights. Also seen that I did not get photos of because they're too fast were a snake that I saw once, and also many rabbits, an excessive amount of rabbits. So as you may know, if any of you have experience with vegetative propagation, these three terms, softwood, semi-hardwood, and hardwood, inform some of the techniques of how to grow plants from cuttings. So I found a lot to learn from these terms, and I put this together to kind of just go over how they're defined, and how to work with these terms instead of being confused by them. So this photo shows one example of how one could define these three terms. These cuttings are likely, I did not take this photo. All the other photos are mine, but this one's just one I just found for this example. These cuttings were likely taken from one plant and just are different parts of the same branch. And as you can see, there's like different densities, different thicknesses, different maturities. And so that's one example of how you can define these terms from one another. However, I find this to be a little bit more of a comprehensive approach to how to think of these terms. So these terms are characteristics of plant type, plant tissue type, and ways to break that down even further is that they might be varying in tissue because of seasonal inference. So is it being cut from a plant in the spring or in the fall when it's dormant? Another way is if it's just a species specific tissue density, not not at each plant is comparable because, as we know, the morphology of plants varies by species. So there's many exceptions to, a more simplified definition like the former slide had. Another way of thinking of it is it's young versus old tissue, and it really depends on the maturity. But it's rare to find a case where one of these categories would apply to all plants. So here's just some photos to kind of show this example. So on the left-hand side, we have something more considerably to be softwood. On the right hand, it's hardwood. And then the things in between, you might consider them semi-hardwood, but there's also some exceptions. Like, for example, this here in the bottom middle, this is coyote brush. And we found this to be most successful from a mallet cutting, which is really a hardwood with softwood coming out of it. And so we would call this a mallet cutting. And so a way to kind of work around these rigid definitions is to apply new terms to it. So there's terms like mallet cuttings, there's terms like tips, and you can use these terms in combination with softwood or hardwood. And then up here, we have blackberry. This is the Rubus ursinus California blackberry. And the stem tissue of blackberry, it really doesn't change that much as it matures. And so, yeah, there's certain species that can be a little bit confusing if the collector is trying to like fit the specific scion or the cutting into one of these definitions. Yeah, and this is important because it's helpful for record keeping, but it's also helpful for interpreting literature. So if there's a propagation protocol that calls for semi-hardwood of something, you want to know what to look for and what to avoid. So here are some more rigorous definitions of these three types. I'm not going to read through these, but this is available to screenshot as a resource if needed. Yeah, so getting into just general vegetative propagation. Here's a drawing of the different materials that you would need to perform vegetative propagation. And most notably, sanitation is one of the most important things. So you see bleach here. We also use rubbing alcohol. Sanitation is really important to having successful cuttings route because you kind of have to treat it like you're giving your friend surgery, because an open wound could get infected and you have the option of either rooting or rotting. Also, this image of these drawings, this is from a small pocket guide that I made for the volunteers. I have a typo on this, so pay no attention to that. But this was one of the deliverables that I put together for this contract in effort to turn some of the propagation protocols that were outlined on spreadsheets and to put them into a more like comprehensive guide and format for volunteers to be able to use. So this right here is an example of one species propagation protocol. So each of these categories right here break down what to look for, when to collect it, the length, which is actually in count by node, and estimated length. But the unit of measurement with vegetative propagation is really the nodes themselves. And then what container to prepare it in, and then more of the qualitative stuff to look for that's unique to each species. Another thing that we found to be really important is not making silly mistakes like planting something upside down. Might seem silly, but there's certain species where you can collect it out in the wild and then you bring it back to the nursery to prepare and pot, and it's actually really difficult to tell up from down. So this is a lot of the time applicable with rhizomes and vines. So they tend to have nodes that don't have much of a direction to them that can inform up from down. So in order to accommodate for this, when collecting, we make a sharp angle cut on the side that is the side of the rhizome or the vine that is pointing farthest from the main shoot, so that we know that that is the point, that's the side that needs to be prepped and potted to point down. With vines, this can be a little bit tricky because, in the wild, there's vines that are crawling all over the place, and sometimes it requires a bit of observational analysis to like track back a vine to see where its central root system is, and that can be tricky. And sometimes it takes something like intuition, I guess. So here's just to show you, this is a rose rhizome, and these little dots here are nodes, but they don't infer much direction. So yeah, that's why we do this. So getting into the genetic diversity and how we manage to maintain that while doing clonal grow outs. We track it all. So here's a map that shows all the different points from parent plants that we've collected from. So each one of these points represents a unique ecotype. So a genetically distinct organism within the same species, which is wild plants with specific traits adapted in response to environmental conditions. So this is essentially a way of saying diversity does not stop at the species level. So we only collected 13 different species, but within that we had over 50 ecotypes to account for, and over 400 individual plants by the end of that. Yeah, ecotypes are a cool thing to think about, and it gets a little trickier than that, and we'll get into that. In the late fall, we were faced with the seasonal flooding, right? And so we decided to take to the waters and collect from boats and create little portable collection kits as well. So that was a lot of fun. So here are some other methods that were developed as one of the deliverables as part of this contract. So the collection methods here are meant to provide for like adaptable methods and decision making. So when you're out in the wild scouting, looking for specific populations, the distribution of those populations will vary. So these different collection strategies, radial, linear, boundary, individual, are meant to be chosen depending on the distribution and how to and a way to collect from multiple individuals in a population while also assuming that there is some genetic diversity in that, and collecting to inform that. Known and unknown diversity is something that we also have to consider, and what that's referring to is whether a plant is growing in the wild via rhizome or seed. So what is its dominant reproductive method when it's out in the wild? And that's when we can sort of assume whether it has unique genetics or if it has the clonal genetics that are present in like the whole population of an area, like a rhizomatic plant would. The PAD rating is another part of the methods that we create, that I created. PAD stands for priority, accessibility, distance, and it's just numbered 1, 2, 3. And this is a rating that's meant to inform future collections and how to prioritize them in order to maintain as much genetic diversity as possible. So, for example, if we're always collecting by the roadside where it's really easy and accessible, we're not going to be we're not getting a representative sample of the genetics of the whole preserve. So in order to prioritize ones that are inaccessible, so top priority being 1, top priority being 1 only accessible by boat, means you have to get a little bit more adventurous to reach some of the more wild and rare genetics. Now we'll get into the community side of things. So this project was affected by the funding freezes, still is, but we're continuing to do this work. We have the ACE interns that are in large part managing the nursery now, and we really leaned into the volunteer side of this actually in response to the first shutdown, both in an effort to to utilize volunteer labor, but also an effort to bring more meaning to this project, and to provide access to this land, and relationship to the land as well. This flyer is for our upcoming ones this fall. So if you would like to attend, we would love to have you there. The last slide also has this flyer too. So if you're running around and can't grab a screenshot of this, no worries. Just wait till the last slide. Here's some more photos from our volunteer collections. So initially, we were doing a hike, collecting plants out in the wild and demonstrating how to do that, and then bringing everything back to the nursery here and preparing using rooting hormone, potting them up. And that's one approach to doing things, and it works well in the fall when hardwood species are nice and dormant and hardy and don't like die quickly through transportation. However, we ended up, I created these mobile kits for propagation. So we combined the 2 steps by bringing out our pots and our bins into the fields with us, and just took cuttings straight from the plant and then prepared it and potted it up right out there in the fields, for efficiency. And it also is a pretty good technique for doing softwood cuttings, because softwood is a lot more, it wilts and dies faster. So the risk of having the transportation time take too long is a risk to having to not having a successful rooting. Now I will get into some of the species specific findings that we had. This is not going to go into every single species or every single like protocol that we tested, but these are just like the takeaways, the most interesting findings I think that we found. So here we have mugwort. Mugwort has very high success rates, and propagates very easily from rhizomes. What I found very cool is just the diversity of the morphology of the young shoots. as you can see in this photo, here's 6 different ecotypes of the one species, Artemisia douglassiana. I think this photo is a good way to kind of to teach as it's a good tool for teaching because it's a visual, it's good for visual learners like myself. Coyote brush was also one that we learned a good deal about. This one, the propagation protocols that I was using in the spreadsheets were pretty vague, and well, they all were, but this species specifically, the way its morphology is made it kind of confusing to decide what a 3-node cutting is, because the nodes on the hardwood mature branches of these are not visible. They're not easy to see. But a mallet cutting is what we found to be the most successful. So here in the middle photo, that's the type of cutting that we found to be the most successful. A lot of cuttings, you have to remove majority of the leaves off of them, but this is an example of one that actually preferred to keep most of its leaves intact and to stay in a humidity chamber for about 3 months before being transferred out. This is also a dioecious plant, so its female and male flowers are grown on separate individual plants. So this is one that it's important to record that at the time of collections, you know which one that you're growing out in the nursery, and then that can then inform the decisions about outplanting. And this is possible because since this is a late season pollinator plant, there's almost always flowers still intact on the plants this time of year. And this right now is the time to take cuttings of Baccharis. So if you see it and you want it, go and get it. Another Baccharis, glutinosa. This one, we got kind of a late start testing on it because I was not informed that we had it on the preserve. But now we have it outplanted. We only have one known individual population of this plant on the preserve. And we found division to be a successful way to propagate it. And we're also trying it currently from rhizomes. And since this is the only individual, it raises the question of, okay, is vegetative propagation the best way to, get more of this plant's presence out on the landscape? Or should we be doing it from seed? So our current plan is to monitor the ones that we're outplanting, and once that has established and started to produce seed, we can then collect the seed from that, and then maybe have a small greenhouse specific to that one need. Oops. Cephalanthus occidentalis, buttonbush. This one also has a very long dormancy period. So this one was one that was easy to start to think that it has not, what am I saying? If something's in the nursery for a long period of time and it hasn't shown much visible growth, it's easy to start losing hope that it's even going to grow. But this is one that surprised us because it eventually did grow. I think we had at that point already tossed some of the cuttings, so unfortunately that was a loss. But this year we know that that's something to pay attention to. This one also loves being waterlogged out in the wild. So once it's rooted, it does really well in pots of native clay soil. And this other photo on the far right is a really funny thing that that's out at the preserve. So this is the same plant. This is a tree, but this is one specific location where whoever's managing it annually chops it down. And so I I have kind of a feeling that this is one individual that has just kind of adapted to that disruption and has started to grow laterally along the ground. It's pretty cool. And I also find the color just wild, the red new growth of it. Elderberry. This is one that there's a lot of existing literature on. I don't know if you guys can see the thing that I keep moving around. Maybe I should just close that. Oh no, what did I do? Sorry, y'all. What happened? Okay, back to elderberry. So yeah, elderberry is known to have really high rooting success year round. It has, I think it's actually similar to willow in that it has a lot of natural rooting hormones and auxin in it. It can be propagated year round. And so knowing this, it kind of raises the question of, well, when is the best time to propagate it? What's the optimal timing and what makes sense along with the rest of our management practices? And I like to think that since you can grow it from hardwood cuttings, that's probably the best way to do it. Since a hardwood cutting is likely to be a lot more adaptable and like durable and more and mature faster than a softwood cutting. It also has pretty fragile root systems, so it's just good to know how to be really gentle when transplanting this plant and also outplanting it. And also, I think for that reason, it's best for it to be grown out in the nursery for at least a year before outplanting. California rose. This is one that we learned a lot about. We did successfully do some air layering on rose, which is kind of outlined here in these photos. It's a technique where you essentially try and convince a plant to grow roots in a secondary location of the plant while it's still growing in the ground, or in this case, in some plugs. And then once roots have grown in this little treated area, you cut the plant and then you got another plant. So yeah, we found this to be successful. But another technique that we found to be even more successful with this species was brush layering. So instead of taking the time and labor of growing this out in a nursery, you can just collect the plant and then directly outplant that cutting in the same day. And we actually found that to be more successful than plants that were grown out at the nursery. And this is brush layering is a technique that we're going to be kind of testing with more than just species that it's frequently used with. Like willow is probably the most common plant that is used with this technique. But now we know that you can do it with rose. I believe that you can probably also do it really easily with most plants that grow via rhizome. Cornus sericea, red twig dogwood. This one we also did some experimental techniques on. This photo here at the bottom is ground layering. So it's essentially the same technique as air layering, except instead of this being suspended in the air, it's pinned to the ground. So then it can access the moisture of the earth throughout the winter and fall. So yeah, we found this to be successful, and this species was also fascinating to see the different ways it would grow. I guess they could be considered morphological differences, but like it was growing in the marsh, it was growing in the understory. There's one growing near the visitor center, and each one of these, unless you knew it, would look like a completely different species. The branching structure was vastly different from one another. So I guess that implies that this is a really adaptable plant to many different environmental conditions. Wild blackberry, California blackberry, Rubus ursinus. This is also a very cool plant. It sends out roots from a node when it's in the presence of moisture. So what that means is that there's water and nutrients kind of going both directions. And so in theory, what that means is that ambiguous node direction is less important to pay attention with this species, but that's something that we would like to actually put to the test. And out in the nursery right now, we're testing mimicking that strategy in the nursery. So basically bending one of these shoots vines over and just allowing it to root naturally in another pot, and then cutting it once it has rooted. It's also pretty sensitive to sun compared to any of the other plants that we grow. And it requires humidity for the establishment of it. So needs to be in one of those plastic humidity chamber bins to be successfully rooted. Also, so this is also Rubus ursinus, California blackberry. It's dioecious, similar to coyote brush. So here's two photos of the difference of the flowers between the female and the male. This one's trickier to know when collecting whether it's male or female, because the flowers are not usually present in the fall. So this is like one of the species that, if we did want to be as vigorously informed with our restoration practices, then we'd probably have to have like a specific mapping project unique to this species, so that we can identify where the different pistillate and staminate plants are around on the properties. And then if you're lucky and it just starts flowering when it's in the bin, like in this photo over here, then you can just record what it is then and there. But this only happened in this one particular case for us. Okay, now getting into some of the more laborious stuff, or infrastructure. This is two photos just to present the progress that we made. We made this progress in just one day. It felt great. It felt like, yeah, between me and the interns, I was convinced that we could build a house after this day. A lot of these materials we sourced from this pile, and a majority of the infrastructure that we built out was just materials found on site. Yeah, and at this moment, we are also aware that some of these are soaked with creosote, these railroad ties. So we're going to be mindful of that when as we're using it. Here's the other side. So as you can see, like this is the retaining wall. And then right on the other side of that, we have this setup. These are all the same view of the same area, but at different times of the year. And this slide is meant to demonstrate the phases of growth. So in early spring, we're still doing some rooting. So like these plants over here, they need mostly shade. And so we kept them over here and kept ones that were already rooted and starting to establish in full sun. And then as spring progressed, and in other areas of the preserve, the canopy is starting to leaf out. This shade cloth, all flowy and holy, to me was kind of a way of trying to mimic the canopy leafing out. And whether or not the plants really appreciated that level of detail, I appreciated it. And I like to think of like the process of growing wild plants as being really a practice in really minimal changes and minimal inputs as well. So we didn't use fertilizers. We had worm castings, and we also did. I've taken some sludge from some ponds and just like fed that into the plants. um, yeah. So yeah, this is like meant to mimic leaf flickering, and then by summer, we had a full shade structure and full irrigation setup so that we could get through the summer. Luckily, as we know, this year was pretty mild. So yeah, it wasn't all that intensive to help the plants limp through summer. Um, so yeah, all this to say, our approach to phasing these plants through their different stages is a little bit more gradual than typical nursery practices. Uh, these three phases here, establishment, rapid growth, and hardening, are ones that are typically considered when using like greenhouses where there's fully controlled conditions, which is like this establishment and rapid growth phase. And then once it's moved out of a greenhouse, then it goes into like the hardening phase. Since our only fully controlled conditions, almost all fully controlled conditions, not temperature, but wind and humidity, was possible to control just within these like humidity chambers here that met our needs for the establishment phase. And then the rest of it was really just like kind of gradually introducing plants to what it is to be out in the wild climate of the preserve. So future directions, we plan to keep this going to some degree. Like I currently don't have any funding for it, but I'm staying involved as much as I can. This is just from last week. We did an outplanting of the slope just beside the nursery, and we're looking to kind of actually restore habitat right there where the nursery is, and kind of go out from there. And it'll also provide a good place for experimenting and monitoring outplanting techniques. Yeah, so I hope that the staff and the interns are really able to to use that as a valuable approach in restoration. So on this day, we had a bunch of fourth graders come out, and I'll say fourth graders are a great way to have the planting distributions randomized, because in restoration, you never want to plant in rows or like really controlled clusters. So yeah, they did a great job. We also have a few burn sites on the preserve, and we're hoping that there's one that we can do an outplanting project on this fall. This was very cool to experience, to witness. If you ever have a chance to attend a controlled burn, I highly recommend it. To me, it was the only way I could describe the feeling of being there is something between attending a parade and sailing. Like you're harnessing the wind, the element of wind. At least you're observing someone else do it. I didn't do it myself. But then you're like walking around the site with a bunch of other people. Yeah, it was special. As I mentioned before, one of the approaches that I took to fertilizing the plants in the nursery is to use native soils. So here's the photo of the one site that I had collected some sludge from. And in theory, these ponds have the mineral composition and the nutrients of the same landscape that these plants will end up in. So nurturing them with the same inputs, I think would be a good approach to growing them. Of course, being mindful of the sustainability of harvesting native materials is important too. This is a whole like this is a whole nother talk here, but this is water hyacinth, and it's currently being mostly managed with herbicide. It does grow on the surface of the water, so this raises red flags to me, since everything in the water is connected. But yeah, this is definitely beyond the scope of this current project, but it's something that I really have some strong curiosities about, because if this plant was managed in a way where its biomass could be used for something useful and potentially even economically beneficial, that to me seems like a smarter approach to managing it than spraying it with herbicide. And there's actually a lot of studies that have been researched in other countries where they don't have as much of the economic and chemical resources that we do. So they've been kind of forced to get creative with managing this plant. And it's been used for all kinds of things, from creating biofuel, biochar, to like paper products or compost, feed for your cattle, you name it, fish food. Anyway, very cool plant. It's one I like to think about and dream big about. But this kind of project would require a major grant. And currently the grant that we applied for is stuck up at a higher level. Anyway, all that to say, thank you to all of these people and agencies for getting me to this point. It's a weird time to be starting to do government funded restoration projects, but yeah, the network of people and mentorship is really what helps us grow. And I'm confident that we'll get through this and hopefully come out on the other side stronger. Here is my last slide. If you actually are inspired and feel like donating to this project, you can call the Nature Conservancy and earmark funds, and then just earmark them to the Cosumnes River Preserve Nursery with my name. These other things here are other ways to get in contact with me, and there are other projects that I'm involved in. But to me, nothing is ever fully isolated from each other. Everything's connected. And I'm always looking to network with people that are interested in, building community and restoring habitat, both in urban and wild landscapes. Finally, this is the flyer once again. So if you'd like to join us for some fall collections this year, come on out. You can register here. You can also just show up the day of, and we'll be happy to accommodate you. Okay, thanks for your time, everyone.
Great. Thank you, Mary. That was great. We do have one question from Amy, and she is asking, why do you find it important to conduct the rooting phase in the shade?
So plants prefer to root in the shade, as long as they, so it's mostly hardwood cuttings and rhizome plants that like shade, especially in their winter dormancy. If there's too much sunlight, it risks drying out the plant. It risks, well, the best way I could actually answer that is that roots grow underground in the dark. So as the very first step, you want the plant to grow roots before it starts growing leaves. And there are some cases where they'll just start growing leaves first. But yeah, to have the highest success, you want like full shade with ambient light.
I would think the other thing too is that if you're rooting in an enclosed container, too much sun is going to heat up the air in that container quite a bit as well, and could potentially cook your newly planted cuttings.
Yeah, definitely.
I had a question. I was wondering, so I love how much thought you're putting into, how you're collecting all of your material, but I was wondering, so what is guiding like what plants you are propagating? Are there specific needs for certain areas of the reserve, or is it just like we're trying to, um, you know, revegetate a certain habitat type? Like, how are you selecting what plants you're propagating um, for the needs of outplanting?
So for our initial contract, I was kind of just starting with what was possible. So looking over the propagation protocols and seeing, okay, we're starting this project now. What is here that I can grow? And what can I start with? And just start testing. Now we're at the point where we need to start knowing that a little bit more intentionally so that we can, have them used for specific projects. So yeah, actually just next week I'm going to have a meeting with the preserve manager, and we're going to go over that and see which plants we should prioritize. And I think this year we're starting. I heard some expressed interest in pipe vine, which we haven't grown yet. So
Yeah, cool. No, yeah, I was curious when you mentioned, that there was only that one plant of the Baccharis glutenosum on the reserve, and like you're targeting that for propagation. I was just wondering, like, oh, it just made me think, if there's only one plant on the reserve, you know, is it because that plant is not really suited for that area, or is it because yeah, like for some reason that plant would be great for, outplanting on more areas, or like does it provide a specific habitat value or something? So yeah, lots of questions.
yeah. And one other thing that informs what we grow and where we plant things is the current management practices. So like
Mm,
right now, there's a lot of sites that are specifically managed for migratory waterfowl. And so like they don't want raptors habitat in certain areas. And so there's a few islands that are actually mowed annually to the ground. And so like there are a bunch of exotics growing on these islands, and we'd like to consider managing that and replacing it with natives, but we'd have to keep it to like non-woody species.
yeah, very cool. I love going out to Cosumnes. Um, it's a pretty big place. So as far as if people wanted to visit the nursery or come out for your volunteer day, um, will that address get them to the nursery or to the right spot?
Yeah, so the address is to the visitor center.
Okay.
Um, yeah, probably where everyone's familiar with if you've been there. The nursery itself it's on it's near the barn, which is not open to the public, but we do allow our volunteers to come to the nursery. So it's something that we would just like caravan over to.
Okay. So meeting at the visitor center
Mhm.
for the work days would be appropriate. Okay.
Yeah,
Great. Thank you.
I had a quick question, if that's okay.
Yeah.
Um,
of
uh,
course.
Related to where you're planting things, Mary, do you ever have to do like site prep or like clearing of stuff before you plant? Or like if there's like a lot of exotics or invasives or something like that, do you have to do a lot of work, or do you try to choose places that are ideal? Um, how does that work?
Yeah, yeah. So there's a lot of battling the invasives. I'm not I'm not the one doing that labor, but yeah, so herbicide use is pretty widespread to control for the invasives, but like for example, the slope that we planted out just last week, which is right next to the nursery, we wanted to make sure there was no herbicides used there. So we solarized the slope through the summer. And there's also HRT restoration days, I believe twice a month on Saturdays. And a majority of their approach to managing it is pulling it by hand.
That's awesome. Dang.
Yeah. One last question that I had was, so ideal planting time is fall and ideal propagation starting time is fall. So are you having a lot of overlap in your nursery, or are you able to get things outplanted in order to make space for the new propagation stuff? Or you have it on two different sides of the nursery?
There's everything's kind of like always getting moved around. And right now, the ACE interns are doing a lot of that work. So yeah, they planted a bunch of stuff. They actually got started kind of early, like kind of late summer,
Mhm.
with some of the trees and stuff. But yeah, there's also like most of the plants that we grow, they don't actually need to stay in the nursery past their rooting.
Okay.
It's just yeah, we didn't have everything in line to get everything outplanted immediately, which is part of the reason this year we want to try doing a lot more with brush layering so that we can really cut out a lot of the labor and the time that it takes for things to grow.
Yeah, for sure. Yeah, and I guess most of the species that you're dealing with are riparian, so maybe there is year-round water for them where you're potentially outplanting.
Yeah, well, the water's all managed and controlled, so it can definitely
Gotcha.
make it make it accessible.
Great. All right. It doesn't look like we have any other questions. I see in the chat, Carol said that it was a delightful, informative project. Good luck with your endeavor. Yeah.
Thanks, Carol.
And I think that is it. So thank you so much for joining us tonight. We apologize for the tech issues last month, and we are glad that we were able to get this done and recorded.
Yeah, so happy to have this opportunity.
Yeah.
No
Thank
worries.
you
I
so
make mistakes
much.
every day, all the time, because I'm a human.
Well, it's the hybrid that's been so problematic for us. It's like if the Zoom isn't working or the room audio or visual isn't working, it's always something. So
Well, the
thank you for
fastest
being
way to learn
accommodating.
is through failure.
Well, it'd be nice if we didn't keep coming up with new ways to fail. So, all right. Thank you very much. Have a good evening, everybody.
Thanks, y'all.
Bye.